Hello, welcome to your lecture on the neurobiology of sex and gender. So, the learning objectives for today are 4 questions that I hope will nicely wrap or shed light on your reading in the Carlson textbook with respect to reproductive behavior. So, in order to consider courtship and mating and parenting, and their neurobiological underpinnings, we first have to consider some questions. The first is, why have sex or sexes that have sex? How do we get sexes, that is, male or female individuals? What is the difference between males and females that can be biologically determined? Is there a difference? And can biology help us understand non binary sex and even the question of gender? So, first of all, why have sex and sexes? The first organisms, back when we originated in a primordial soup, were females that asexually reproduced through a process of cloning, similar to what bacteria do today, where mother cells divide into daughter cells, which divide into granddaughter cells, and so on and so forth. The important thing about animals that clone is that they are identical. They lack genetic variation. So bacteria have evolved certain methods for stealing new DNA to help enrichen their molecular makeup and make it less likely that a single, for instance, antibacterial agent can wipe them out. But generally speaking, being a clone or descended from clones, being deeply inbred, is not necessarily a good thing. For instance, the dinosaurs were thought to have become canalized, have become so genetically fixed that they lacked the capacity to adapt to changing environmental circumstances. So there are good things to come from having genetic variation. So genetic variation, how do you get that? You get it through genetic recombination. That is having DNA that comes from two parents that is shuffled before it is joined in a shared offspring. And that genetic recombination confers hybrid vigor. It means that when genes are shuffled between generations and recombined, there becomes more opportunity for adaptation, for novel physiological and behavioral responses to emerge in the face of novel environmental circumstances or environmental changes. So the idea of sex or sexual reproduction is something that has evolved because of the benefits of genetic recombination and hybrid vigor in a number of species. And in this table, from a different textbook, you can see that there's a wide array of animals, including invertebrates and fishes and birds and mammals that have developed some form of sexual reproduction, right? And in the case of some invertebrates, they have organisms, individuals that have both male and female parts that are hermaphrodites. And that's how they accomplish genetic recombination and some semblance of hybrid vigor. In fishes, we have some species that will be female at one life stage or in certain population circumstances, and will convert to a different sex depending on ecological factors. Obviously, in birds and mammals, we have two sexes. The difference is that in many mammals, we tend to have very dimorphic sexes, meaning that males and females take forms, have phenotypes that are drastically different. Monomorphic species are birds, for instance, or animals where it's hard to, with the naked eye, differentiate male and female members. So lots of animals have figured out a way to have sex so as to, develop, experience genetic recombination that leads to hybrid vigor. How they've done that has been determined by different gene systems. So we humans obviously have XX and Y, XY. The male determining genetic system varies across the animal kingdom. In some non-primate mammals, it's UBE1 is the equivalent of the Y chromosome gene. Moles have lost the Y chromosome entirely, leaving both sexes with either an unpaired X chromosome or both females and males with XX in some species. Birds and some insects use the WZ sex determination system. And in those species, females are the heterogametic sex, meaning they are the ones that have two different kinds of sex chromosomes. Males have the same kind of sex chromosome. So humans, males are the heterogametic sex. We have XY males and XX females. The haplodiploid system is found in ants, bees, and wasps. And in those species, males are um, have one set of genes, one complement of genes, and females have a double complement of genes. So temperature is another way that maleness is determined. In this instance, in alligators and in some reptile species. And then I mentioned to you in the previous slide that there are several species, including invertebrates like snails and earthworms and some insects, that will change sex or have true hermaphrodites, where reproduction may occur in a way that is wholly unfamiliar to us, for instance, by parthenogenesis, and or infection in the case of some species. So we've talked about the genetic determination of sex, and why we would want to have genetic recombination in order to get hybrid vigor. So why are there two sexes as opposed to 3 or 4, you might be wondering, or even one individual that is a hermaphrodite? So the basic mammalian body type, as you will recall from the first slide where I talked about the first species, the first examples of life being female, is still female. So the question is, how do we get something other than female, something that is partly female in the case of real hermaphrodites? But ultimately, how does the plan to be female get thrown off course to produce, dare we say it, a male counterpart? So how do we get from this? In the case of humans, the female karyotype is 46XX. And what that means is that we have 46 autosomes and then 2 X chromosomes. So half of these chromosomes would come from the father and half would come from the mother. And each parent gives one sex chromosome. In this case, um, uh, each parent gave an X chromosome, which resulted in a 46XX female offspring. So how do we get from this to this, where you have 46 odd chromosomes and XY, basically a male? And this is kind of a joke slide. You can see the Y chromosome is mapped out here, and there are things on it like air guitar, spitting, total lack of recall for dates, selective hearing loss. But the important thing that isn't a joke is the testes determining factor, and we'll get to that later on. In the early 90s, so about the time, well, I think before you were born, students in physiological psychology, there was a lot of effort to try to figure out exactly what part of the Y chromosome led to the development of maleness. Over a series of studies, female mice were had their genetics tampered with, and increasingly small stretches of Y chromosomal DNA injected into their um, cell nuclei. And as a result, we were able to identify precisely what part of the Y chromosome leads to maleness. And in 91, Koopman discovered that it was the sex determining region of the Y chromosome, that adding just that small bit of DNA to uh, um, sex cells in mice was sufficient to lead to the development of male sex in otherwise female embryos. And this is the timeline of leading to the discovery of SRY. So you can see science doesn't happen overnight. Starting in 1959, researchers honed in on precisely that tiny stretch of DNA that really makes the difference. So this sex determining region of the Y chromosome is responsible for producing testes determining factor, which is that thing that causes a deviation from the female default to the development of maleness in an embryo. So what sex differences are biologically based? Um, it's important to recognize that sex is um, very complex, that it's multiply determined, and that there can be incoherence or conflict at different levels of sex determination. So biological sex can be both definite and in disagreement with other, at various levels, and also with ultimate gender and sexual orientation in, traditional terms. So chromosomal sex is what we've been talking about until now. And that has to do with whether you have an X and a Y, or an XX, whether you're genetically male or female, or more particularly, whether your Y chromosome actually has the important bit of DNA that's necessary. You could be chromosomally female and chromosomally male and XX if you have a tiny piece of SRY attached to one of your X chromosomes. That has happened. So chromosomal sex has to do with what DNA you have. Gonadal sex has to do with your reproductive organs, your testes or ovaries, your penis or vagina, um, and um, also to some extent reproductive accessory organs like the breasts that function in parental care in the case of mammals by lactating to support one's young. So gonadal sex is a level above the chromosomal sex. Um, Even before one sexually matures, you have, in most instances, identifiable male or female gonadal sexual parts. Hormonal sex has to do with the quantities um, of and the quality of neuroendocrine processes in the body. So, there isn't a set amount of testosterone that makes you male. In fact, testosterone is a precursor for um, important steroids that support gestation and sort of functioning of females as well. So everyone has testosterone. It's the balance of testosterone, the balance of male and female hormones, their receptor systems, and the distribution of those receptor systems and enzymatic factors in body tissues that really leads to the determination of morphological sex as we function physically as reproductively viable male or females. Um, and it'll become more apparent as this lecture progresses how you can have disagreement or conflict at these stages, leading to differences ultimately in behavioral sex. So, sexual differentiation is the process by which we go from being um, a fertilized egg that has XY or XX to being fully male or female at birth. And it looks like this in the case of males. Um, and XY is the starting point. If you have SRY, then you will early in fetal development develop anti-Müllerian hormone and testosterone. And the anti-Müllerian hormone will have the effect of causing the Müllerian ducts to regress. The testosterone will have the effect of causing Wolffian ducts to form, the epididymis, the vas deferens, the seminal vesicles, and other tissues that support the production of sperm down the line. Some of that testosterone will be converted by an enzyme called 5-alpha reductase, and that will lead to the differentiation of external features that help you become identified as male. That 5-alpha reductase turns testosterone into its more potent form, dihydrotestosterone, which causes the formation of the scrotum and the penis. So these are the steps that are required to shift away from the female default to maleness. If you are missing any of the events in this cascade, you will develop along the female default. So testosterone is important for both brain and body masculinization, but it's important to note that testosterone converted by 5-alpha reductase to dihydrotestosterone changes the female default, the body of the developing embryo, to a male body. And yet testosterone converted to estrogen by the enzyme aromatase converts the body plan, the brain plan, of an embryo to a masculinized brain. So the precursor to physical sex and brain sex differences is testosterone, but in the case of the body, it's testosterone converted to dihydrotestosterone by 5-alpha reductase. In the case of the brain, it's testosterone converted to estrogen by aromatase. So early in development, hormones have an effect on organizing the body and the brain. They organize the genitals, they organize the important brain areas that coordinate sex typical behavior, often behaviors related to mating, courtship, um, and parenting, as well as reproductive competition. So there's this organizational effect, and then we are born, and there are very little differences in terms of the levels of circulating hormones in young boys and girls. But then as they approach puberty, we have another surge of hormones that interact with the underlying tissues that were formed in utero, in the womb, causing a dramatic shift, dramatic physical changes in boys and girls that metamorphosize essentially into young men and women. Um, and as part of this activational phase, um, it it's also important to recognize that there are activational effects of hormones as they are secreted. The sexually differentiated behaviors are triggered by hormones interacting on this neurological landscape that was initially organized in utero. So in phase one, embryonic development from the female default leads to organization of male or female genitalia. And then in phase two, with adolescence and puberty, we have the formation of secondary sex characteristics. And secondary sex characteristics are those things that make us dimorphic, differences that emerge in adolescence that are activated by the introduction of estrogen and testosterone that lead to enlarged body size in males, particularly denser muscle mass, the characteristic differences, the V torso in males and the hourglass figure in females, with a larger hip and breast to waist ratio, differences in hair distribution, fat deposition, greater muscle mass in males, more fat deposited in the breasts and the hips in females, also differences in behavior and brain functioning. It's probably no surprise to you that males are more likely to commit homicide, and that particularly younger males are more likely to commit homicide than males in their 40s and 50s and beyond. So the activational effects of hormones, particularly in youth, are very pronounced. And it's important to note that they differ. So hormones differ in the extent to which they activate sex typical behaviors that help with mating versus those that help with parenting. So hormones that are circulated activate um, and suppress um, opposing behaviors. So being on the prowl for a mate is in direct opposition with parenting, where you're investing in a, a bonded partnership, and focusing on existing young as opposed to the focus on creating new young. And so it shouldn't surprise you that when animals are seeking new mating opportunities, their testosterone levels are higher. They're also much more um, competitive, particularly against same sex individuals. So here you see two baboons fighting on the left hand side. By contrast, when animals are in parenting and bonding mode, there's a general tendency to have lower levels of testosterone and higher levels of oxytocin and vasopressin, which are chemicals that are produced that help cement the bond between parents and the bonds between parents and children, sort of reinforcing the um, the drive to create and maintain a family. So normal development of the external genitalia exists, and to some extent the endocrine profile, how much testosterone or estrogen you have, exists on a continuum. So on one end is extreme masculinity, on the other end is extreme femininity. Most people are somewhere along this continuum, with very few people being either immediately in between, or rather exactly in between, or on one or the other extreme end. And this is because the biosynthetic processes underpinning um, uh, the neuroendocrine functioning of males and females are so complicated. So you can see here, everything starts with cholesterol. And so that's why you need to have some fat in your diet. But at every stage, in order to get the stress hormone cortisol, which you see here, or estrogens, um, you need to have some enzyme, right, that converts that starting steroid um, compound or molecule to uh, subsequent products, right? And you can see how at any stage, if you're lacking this enzyme, or you have an accumulation of one thing at one stage in the production chain, the manufacturing of the end products will vary. So in order to get from cholesterol to estradiol, there are all these stages, including precursor steroids and enzymes that have to unfold. And you can see that from estradiol, to get to estradiol, you need to convert testosterone via aromatase. And so having no aromatase could mean that um, you lack the necessary um, estrogen, this potent estrogen, to properly function as female, or in the case of males whose brains are actually um, masculinized by estradiol, to have a properly masculine, and by proper I mean as the system is designed, a fully masculinized brain. Okay, so we have a number of scenarios whereby known genetic anomalies lead to abnormal sex development, and two that are mentioned in your textbook and probably aren't unfamiliar to you include Turner syndrome, which affects 1 in 5,000 women, and Klinefelter syndrome, which affects 1 in 700 men. In the case of Turner syndrome, you end up with 45 XO, so you're missing one sex chromosome. And so you have almost an insufficient dose of X chromosome to fully manifest as a female. And with Klinefelter's, you have an extra X chromosome. So you're male, but you have a double dose of X chromosome as a male, and that has the effect of leading to certain prototypical changes. In the case of Turner's females, you will notice that they have, you know, shorter stature, based on this illustration, a shielded chest, webbed neck. They, by virtue of lacking sufficient dosage of the X chromosome, will have underdeveloped breasts and rudimentary ovaries. In the case of Klinefelter's males, they, by virtue of having an extra X chromosome and more dosage, a higher dose of what that brings, they will have poor beard growth, a female pattern of pubic hair, testicular atrophy, meaning sort of abnormally small testicles. They also tend to have taller stature and minor breast development and poor beard growth. So some babies are genetically XX or XY. They're not Turners or Klinefelters, but are born with uncertain phenotypic, meaning external or mismatched sex. And often it has to do with either the problem with some endogenous biochemical pathway that's required for sexual differentiation, or the introduction of prescriptions or chemicals from the environment, exogenous substances that alter sexual differentiation from the typical pattern. And four are going to be discussed either in this lecture or in your book. They include congenital adrenal hyperplasia, androgen insensitivity, 5-alpha reductase deficiency, and maternal progesterone prescription. So congenital adrenal hyperplasia is what happens when a baby is missing 21 hydroxylase. And so progesterone that is supposed to be converted down this pathway builds up over here, and some of it ends up flowing into the pathway, culminating in testosterone. And so what ends up happening, I mean, think of this as like a plumbing system, is that if you don't have this enzyme, you end up with more testosterone. And if you happen to be an XX baby, then you'll have higher levels of testosterone, of androgens, and that is associated with having, when you're born, more ambiguous sex, so an enlarged clitoris in this case. And then, as is the case in retrospective studies, many people suffering congenital adrenal hyperplasia will say that they, although being assigned female identity at birth, felt like tomboys, felt male, In studies, they have been compared with botched circumcision um, subjects, and as a sort of control um, for the effects of having uh, androgen on board, um, that leads you to experience life as a male, and being forced in both circumstances, either through surgical mistake or treatment purely on the basis of ambiguous sex, how that relates to actual experience as male, or in the case of these individuals, as females, um, who are raised as females but identify as males. Another example is androgen insensitivity syndrome. And in this instance, we have individuals that are XY and have SRY will develop normal testes, but because their androgen receptors are defective, they basically have no way, regardless of how much androgen is produced, of activating necessarily the effects of testosterone. So imagine the signal exists, so the testosterone exists, but your receptor that is responsible for binding that testosterone is defective. So no amount of testosterone is going to lead to the effective differentiation. So the female default is maintained, and these individuals manifest as female, although they may have some rudimentary male organs that developed before testosterone came on board. Maybe they had enough testes determining factor, but led to, in the absence of testosterone, perhaps the identification as female upon birth. 5-alpha reductase deficiency is what happens when an XY individual develops as a female because they can't convert testosterone to the dihydrotestosterone that's responsible for masculinizing the body. You'll remember me saying earlier in this lecture that testosterone comes from cholesterol, and that 5-alpha reductase converts testosterone to dihydrotestosterone, which differentiates the body, and that aromatase converts testosterone to estradiol, which differentiates the brain. And so if you don't have 5-alpha reductase, your body will manifest like that of the female default. And yet you will have an XY, and um, particularly at puberty, you will start to produce other androgens that will lead to a subtle metamorphosis. And that is essentially what we see with the guevedoches, the Dominican pseudohermaphrodites, that are illustrated in the documentary segment that I have posted on Moodle. In the case of these individuals, they are identified as female at birth, but then as sufficient androgen is produced by other body tissues, um, and sufficiently high levels of testosterone are produced. In adolescence, you can have activation of sex differences without the necessary step of organization. And so these individuals will go from being raised as females to, in many instances, deciding to adopt a male sexual identity and a uh, uh, orient themselves heterosexually uh, choosing female partners for mating purposes. So, can biology explain fluidity or non-binary sex, and where does gender fit in? Um, so, I think what you should know by now, uh, based on this lecture, and your reading, is that sexual differentiation and subsequent gender identity is complicated. That the basic mammalian body plan is female, that SRY gene on the Y chromosome leads to the production of testes determining factor, that the testes produce testosterone, dihydrotestosterone, Mullerian inhibiting factor, which lead to male anatomy. Again, in the absence of all this, you stay with the female default. Testosterone later in gestation is converted to estrogen, and that leads to brain changes that are characteristically male. So there are prenatal organizational effects. But then gender identity isn't neutral at birth. Um, we have um, this sort of genetic and endocrine backdrop that is foundational. And then we have experience in terms of how we are assigned sex at birth and what that means in terms of the reactions of others and how they treat us as male or female or something in between. Um, and then in puberty, we have further changes based on the activational effect of hormones that are secreting, combined with societal factors, right? So, sex assignment at birth, early gender identity um, emerges at 3 to 4 years of age, a little bit before biographical memory, even. And then ultimately, we see that social factors um, interact with our bodily makeup to drive the differentiation of adult gender identity, which often is constrained by cultural or social norms. So you are probably thinking, what are some behavioral differences between males and females? I mentioned the differences in rates of homicide, with males being more homicidal or more likely to commit homicide than women. So we know that the brain plays a role in aggression. The important thing to know is that the brain, that having testosterone isn't poison. It doesn't make you commit homicide. But on the landscape of a male cerebrum, testosterone may more likely trigger homicide than it would do based on the different organization of a female cerebrum. But both males and females are aggressive. They are aggressive in different ways, partly due to cultural norms. So girls are often more verbally aggressive, whereas boys may be more likely to engage in physical aggression that we would describe as violent behavior. So some of the sex differences in the brain correspond with what some people reason may be traditional roles. So in many species, we know, for instance, that um, uh, males will defend territories that will need to be patrolled, whereas females may be a little bit more confined to the nest. In sexually reproducing species that particularly have some dimorphism. And so it shouldn't surprise us that when it comes to cognitive tasks, there's some evidence that males are more adept in tasks involving spatial orientation. Um, Females are better at recalling the location of objects. So there's that difference that may be attributed to our sort of legacy in terms of having gendered or specific roles in terms of our contribution in the in the way of mating, courtship, and parenting. But if you look at this table, what you should see is that many of the traits that are um, attributed um, are particularly excellent in one sex or another are actually quite complementary. So perceptual speed is greater in females. Um, Visualization is better in males. Those are complementary skills. Look at mathematical reasoning. So solving a novel mathematical problem is one aspect of IQ that favors men, but numerical calculation, adding, subtracting, and doing operations with numbers is a particular strength of females. So these skills are very complementary, and that is an important point. So when we talk about sex differences in the brain, there are some areas that have been identified as different volumetrically. Um, the hypothalamus, the thalamus, and the corpus callosum are areas where we've seen differences in the size or the number of nuclei when men and women are compared. Also, the shape, the form, of the corpus callosum, the splenium, a particular region, subregion, is more bulbous in women. The entire callosum shrinks with age in men, but not women. So some differences in terms of the shape, the volume, the number of nuclei, um, or in the case of the caudate, which you see illustrated here in the amygdala, subcortical volumetric differences have even been attributed to um, um, differences in genetic sex. So, for instance, in this study, comparing 46XX females with 46XY males and Klinefelter's males, you can see that the typical males have right and left amygdala sizes that are slightly larger than females, and that females even have right and left amygdalar volumes that are larger than Klinefelter's males. Um, so going along this table, there are a number of differences that you can see. So for instance, Klinefelter's males have caudate volumes that are intermediate between XX females and XY males. In your textbook, there is a lot of ink given to the sexually dimorphic nucleus of the preoptic area. So this is the part of the rat brain that has been intensely scrutinized with respect to sex differences. And you can see here, based on this illustration, that the cell density is greater in the male than it is in the female with respect to the sexually dimorphic nucleus of the preoptic area. And then in the human brain, this is an illustration from your textbook, you can start to imagine based on the varying kinds of data um, that have been put into this sort of computer simulation, how structures may be different in male brains relative to the size of the male brain um, than in female brains. So male brains, because they come from male bodies that are slightly larger based on our species sexual dimorphisms, tend to be larger too. But when you control for body size of the subject, you find that even then, there are some structures that are larger in healthy female brains um, or larger in healthy male brains relative to the opposite sex. So around the millennium, there were some interesting studies by Simon LeVay, a neuroscientist, that showed that there's certain parts of the human brain, the hypothalamus, certain nuclei of the hypothalamus that are not only corresponding to the sexually dimorphic nucleus of the preoptic area in rats, but specifically sexually divergent in male and female humans, and um, somehow related to sexual orientation, such that women and gay men have nuclei of the hypothalamus that are more similar in terms of their cell densities um, relative to heterosexual men. So this was the beginning of our collective understanding or awareness of how differences in brain sex can lead to um, maybe a combination of um, divergence in terms of sexual desire and who you identify as attractive, but also in a sort of disconnect between genetic sex and what kinds of sexually reproductive organs you have, and the target of your sexual desire and subsequent reproductive strategies. So normal development of accessory sex organs includes the brain, and it's hard to you know ascertain or identify a perfectly male brain or a perfectly female brain, just as it's hard to pinpoint a perfectly female or perfectly male body based on the complicated biosynthetic pathways that underpin our endocrine profiles. So there is a, a large area of overlap between what makes us male and female in the body as well as in the brain. And it's very, very complicated. And we're only now beginning to tease apart this very delicate process by which the body and the brain are masculinized.